So I tell us all about her adventures with microbes. So please take it away.
All right. So hi, my name is Kunmanee Buphamanee, but I go by Mac. So today I'm really excited to share my summer research rotation, not far at UW with School of Oceanography with Professor Robert Morris. And yes, basically this is what I work during the summer is to cultivating and sequencing microbial community from oxygen minimum zones and share some insights about the mixotrophic isotope. And why would I care about oxygen minimum zones? And I want to share a little bit on that, on my motivation, because for us as an astrobiologist, we want to look for an analog to a different planetary world, or in my case, is early ocean. So that could be get you interested about this project a little bit. And yes. So, oops. All right. So the motivation about oxygen minimum zone. So mainly my research is focused on utilizing stable isotope system to study biogeochemical cycle in the early Earth environment and to understand how these processes facilitate the evolution of life. And for early Earth ocean, we categorize broadly into 3 stages. So first stage in Archean, basically the ocean is enriched in iron, but for Proterozoic, it's shifted to more euxenics, or basically rich in sulfide species. And then in Phanerozoic, the ocean shifted to more oxygen-rich, or just like modern ocean. So oxygen minimum zones, how we see, has a low oxygen concentration and rich in sulfide concentration, which can be the chemical composition kind of resemble the mid-Proterozoic ocean. So that's why as a geologist and astrobiologist, we want to use OMC as a modern analog of early ocean. And here is the global map of oxygen concentration at 300-meter depth, and I'll just call mid-depth from now on. And you can see that the red color depicts the area where it has high oxygen concentration, and the purple kind of depict the region where oxygen is low. And these areas are classified as OMC, oxygen minimum zone. And it's kind of concentrated around the equator, around the western side of the continent. So yes, so as a geologist, we study rock. Sometimes we may overlook the role of microbial community in the oxygen minimum zone. So that's why this kind of lead me to reach out to Professor Bob Morris. And Bob from UW School of Oceanography, his lab is able to isolate several strains of bacterial sub-O5 clade. And I believe he's probably the only group so far. You can correct me. But yeah, sub-O5 is a microbial community that dominate oxygen minimum zones, and some in fjord, in close sea, and also deep sea hydrothermal system. And this clade is a biological driver of elemental cycling in the mesopelagic zone, because it has metabolic flexibility that can do autotrophic and heterotrophic in the area. So here is the example of sub-O5 clade, which is Candidus globus, and it has a remarkable flexibility because it can store elemental sulfur inside a body to support their growth. So in the environment where sulfur is enriched, they store elemental sulfur. So when they move outside the area where sulfur is low, they can use that sulfur to support the growth. So this metabolic flexibility kind of offer advantage in the area where a chemical concentration was shifting. So yeah, building on this, his research, I'm interested in microbial community in the oxygen minimum zone. So that's why I reached out to him and tried to refine a project. And during that time, Bob has a collaborator, Professor Timothy Mattis, just called Tim, and he's on sabbatical. He's from University of Iowa, looking at a microbe also at that time. And Tim has been studying the metabolic flexibility of sub-O5 under low oxygen minimum zone. low oxygen concentration. And at that time, he wanted to search for sub-O5 in different body. So he went to collect seawater sample from Namibian coast, which is a part of Benguela upwelling system, also an oxygen minimum zone. So he collects seawater from there, and the goal is to identify who live in there. And yeah, the seawater was collected at mid-depth also, and mixed with gristle stock and frozen with liquid nitrogen, and then shift to CTAC and then to UW for the experiment. And that is basically what I'm helping him basically try to identify who live in there. So here is a simplified method of the project. So we have a seawater sample that has been collected and preserved. Now we want to isolate microbe to like a single species, genus, and then we try to grow in a large biomass, a lot of cell density, so we can extract a DNA of the PO microorganism. So this is like the overall project during the summer. So yeah, in detail, now we have seawater, and these seawater will be thawed and then subject to a dilution to extinction processes. So basically, the seawater will progressively dilute it until a single microorganism is stay in the individual well. So after a while, some will grow, some will not make it. So from this micro well plate, I think each well is about like 1.5 mil, so it's pretty small. And we have multiple well. So we collect the microbe that are able to grow to high density, and then we'll transfer it into a larger volume to produce more biomass. We culture it in a 1 liter filtered seawater from Puget Sound and enrich it with thiosulfates to support sulfur oxidizing bacteria. And overall, I think when we got there, we have like 51 culture of microorganism. And so the next step is to identify who are they. We use a whole genome long-read DNA sequencing to identify those microbes. So long-read sequencing offers several advantages because here an example why we use long-read DNA sequencing rather than traditional short-read sequencing. So long-read is like a long fragment of DNA. So that is a key advantage because you give a larger span of DNA. With multiple long span of DNA, it's often easy assembly because you have multiple overlapping. And when we get the full DNA, it's have like a minimal gap. That's from what I understand compared to a short read one. And there is also improved the analysis of repetitive regions because you have multiple, so you can do more of it. And the last thing is with the multiple regions, you can detect the structural variation in that DNA also. So analogy of it, think about like a jigsaw. I think I pulled it from the website, and long read DNA is like a 9-piece puzzle, while short read is like a 900-piece puzzle. So it's easier to complete a 9 pieces jigsaw when it's had more information compared to a short read one. So that's why we use long read DNA sequencing to complete a whole genome. So now the actual work. To do this, we already have a culture in 1 liter volume, and to do that, have to collected bacteria and filter it, concentrate it on the filter. So whatever I have on this filter, it's just bacteria in the culture. Then I'll have to extract just the DNA of it. So this is just like a simple graphic that I can find online. And the process of doing this is, well, we'll like the cell, break it apart, put an enzyme to get rid of the RNA, and then whatever is DNA is gonna stuck on the filter, and we have to press and wash multiple times to remove the seawater and also the impurity of that. And then we clean up the DNA on the filter by dilute it, well, by put it in a solution again, and then it's gonna be a DNA impurity. And then we're trying to verify the DNA out of that solution by using a magnetic bead that will bind with the DNA. And then we'll separate it by put magnetic in it, or just by just the DNA, pull the magnet out, and then the DNA will be dissolved in the solution, and now we get a pure DNA. And I've struggled a lot of these, a long extensive method. It took about 2 days, but there's a lot of steps that could be mistake, and I did some of this with cause like my DNA has a high contaminant in that. So yeah, it's very delicate step. And here is the magnet rack we use to purify DNA out of the solution. And now we have DNA. Now this DNA is ready for sequencing. And the process is called library preparation. And basically, this is the overview protocol for nanopore sequencing. To prep a DNA, we have a DNA in a tube, like a 1 liter tube, 1 milliliter tube. We add an enzyme, which is called transposon something. And this enzyme will cut the DNA into pieces. And this enzyme also has sequences that also insert into the DNA that we cut. It's like we tag the DNA that we cut. So this is like called tagged DNA. So now we have DNA that has been cut and has been tagged. We're gonna add the rapid adapter, the RA. This adapter acts like a carrier, and it will attach to the DNA end, and it will carry the DNA into the sequencing pore. So after I have prepared all of this, these are just like multiple tube, a bunch of clear liquid. The differences is different color in the label. I promise I did not mess up anything, but I don't know. It's just like transfer small liquid from one to the other, and really small, like microliter. So yeah, hard to imagine at that time. And then I transfer all this liquid into the sequencing device. So we use Nanopore MinION, and it has a MinION software. And this device is about like the size of my palm, very portable, very powerful, and it takes about 24 hours to do the sequencing for one single culture. Yeah. So next step is, how does this machine work? This device, basically. So DNA sequencing, the overall idea is the DNA that passing through the pore will disrupt the signal and read to the base. So I will explain that in detail. So first, we have DNA that attached to the rapid adapter. It's DNA and adapter, and this adapter carry it to the pore. And the pore is basically in a membrane that's soaked in salt solution. So when electrical potential apply, this salt, as it's moved through, it will create. It basically has electric in there when it's moving, right? But when adapter carried it in the DNA and passing through the pore, it's disrupt the electrical signal. So as each base, ATCG has different electrical potential passing through the pore, it will disrupt the signal at different value. So that's how they read the disruption of electrical potential and translate it into base ATTCT. So yes, the protocol require about 50 to 70 nanogram of DNA, but I think since we struggle with some strain doesn't grow that much, we put lower down to like 20, and we still get some data from it. So now, after we have a fragment of long-read DNA, now we want to assemble it into one whole genome, try to identify who are they. This is what I got come out from the sequencing device, which is a long grid of DNA fragment. And now I'm trying to combine it into one single whole genome. To do that, I didn't do it myself. We have assembly tool, which is a computer. This is the Galaxy free software online that you can just upload the file and it will assemble for you. So I use Fire, which is a symbol tool for a single molecule sequencing read. And then I have to polishing using Medaka consensus pipeline, which is kind of designed for this nanopores. And what I get out of it is like a whole long genome, something like this. Sometimes it's come out nicely and it show like one contact, like one whole full assembly genome. Sometimes it doesn't, it's give like a fragment and we still can be able to work with that. But I think most of the time we get like a nice one contact. Sometimes it's give two, which is suggests like it's a contaminant in that culture. So now we have the output data of one line whole genome. Then we need to annotate it, try to find who is it. So I used a DFAS, which is also a free software, free server, something. And then this just upload the file and it probably take like 10 to 30 minutes to get it fully read. And it will tell the genomic information, like how many base or how about my, like, how clean is my data? And if it's lucky, it will give me the name of who is it based on the database that it has. And so most of the time it's gave nicely, but sometimes when I put like my DNA is not clean, it's fragment, sometimes it's just like give a bunch of name that we're not sure what it is. So yeah, that is all the work that I've done in like hands-on experiment. And yeah, we get the identification. So overall, out of 51 culture, we 8 of them are not grow in a larger volume for some reason. We have 11 of them ready, but not yet sequences. And then we have 32 sequences and identify who they are. And from that 32 culture, we identify like all of it, and 11 of them are pelagic bacteria, which is like the largest population you see here. And the pelagic bacteria are silent bacteria, which is the most abundant phytoplankton in the ocean, which is kind of makes sense. And these were, I mean, I'm a geologist, I don't really know who they are, but we're doing something in there. And we have pseudo-thioglobus that can do sulfur oxidation, but not exactly sub-O5 that Bob were looking for. So yeah, that is the work. But like I said, some come out nicely and some I have to dig it through, like who they are. And these UBA 868 are the give me a hard time because it takes so long to grow, and I believe like 2 months to grow. And when we harvest it, the cell density does not grow that high, and it's kind of reached the threshold that we like, should we collect it or should we not collect it? And when we try to identify it, it doesn't give me the name right away. I have to get the fragment of it to look in the literature and try to find who it is. So I spent a lot of time like looking at this one, and I think it's given me a good reason that like I should appreciate it. So yeah, so UBA 868, it's a really cool one because it's a mixotroph, and there's only one literature that published about this strain, and this strain dominate in mesopelagic zone, which is about 200 to 1,000 meters where sunlight is limited. They call it a sunlight zone. And all the data coming from, I believe, genomic, and they're trying to compile from the gene library and match it whether it's this group or not, and that's how they identify the abundance. And most of it are just in the deeper ocean or maybe deeper. So yeah, they're abundant in deep ocean. And not only that, is what they do are also important, because the UBA 868 can fix carbon dioxide using energy generated from sulfur oxidation, but it can also take up organic substance from phytoplankton and zooplankton, using these for energy production. So in current research right now, we believe that to regulate it carbon cycle, the player are either autotrophic or heterotrophic, but these microbes can do both. So these metabolic flexibility kind of allow them to thrive in this extreme environment and help regulate it carbon cycle organic material in the deep ocean where nutrient is limited. So it's really a cool player, and it's kind of extend the ecological niche for itself too. And it seemed like a lot of bacteria in oxygen minimum zone, they kind of have these flexibility to do it. So yeah, so summary, with these genomic analysis help us uncover who microbes are and what they do. And another thing I think might be interested for people who interested in technology, these nanopore device sequencing could be applied for future exploration. And what did I learn about metabolic flexibility of microbe in the OMC? Not only provide just like it has evolutionary advantage that allow them to thrive in the difficult environment. And that is very important for us to think about life on other planet or extreme environment. And these mixotrophic bacteria, UBA 868, can be used as model organism to study metabolic flexibility and the regulation of carbon cycle in a diverse environment. And Bob has microbes in his freezer, so everyone is welcome to use, I guess. And the diverse environment is not only mesopelagic ocean, as I'm interested because it's a sulfide-rich water in low oxygen, but another analog for planetary people is the planetary sub-ice ocean, where sunlight cannot reach down and it can still get oxygen from a reaction from a radioactive. Yeah. So that is the overall of my rotation. But of course, I'm also want to share some stuff. So Bob was kindly asked me to like join him, and we get on the 5-day cruise to collect the seawater. Those are the Nissan that collecting water. And I think the deep that we can go is about 200 meters. That is like general depth of Puget Sound, not that deep compared to the ocean. And yeah, we also made a lot of mistakes. First time prepping this Niskin, forgets to close that, so all the water leak out, kind of upset the captain a little bit, that we have to do it again. But yeah, not my fault, there's a multiple too, so. But also, like, later half of the trip, we get a sunny sky, so that it's my office in the back of the ship, trying to do some work collecting different type of microorganism in the seawater. And we do a bunch of filter we use a big pore filter to collect phytoplankton, and here are some we see. We have microscope on board, we get to look at those. And whatever passing through that is just small bacteria, whatever passing through it, it just bacteria and a fragment of it. And I believe Dylan's gonna do the downstream analysis update. He's gonna take over that. I'm just here chilling, collecting data. So beside that, we also have a setup movie at the back of the ship. Very nice. And just nice meal, coffee, and also a free trip on the Deception Passport, which is pretty cool. So yeah. And I would like to thank Bob, Professor Robert Morris from School of Oceanography, that kindly asked, well, not asked, I asked him to join and doing research with him, which is pretty cool. And then we have Tim, Professor Timothy Mattis, that teaching me how to do genomic sequencing and kindly showing me around. And then, of course, Mike Sadler right here. I have to say, I struggle a lot with DNA extraction and purification, and Mike stepped in to help me get through it and get all those nice data. And then we have research vessel, Rachel Carson crew take us to there. Professor Benjamin No, he's a part of the research on the Rachel Carson trip, looking at the fakes and also how virus affected in seawater. And his student, we have Jacob, Sarah, Madeline, Naomi, and Kali on board. And that is him showing me around the lab. So yeah, that's my summer here.
Thank you very much, Matt. You have time for questions.
Yes.
Do you know what UBA 868 has for carbon fixation knowledge?
Oh, the question is, do I know if UBA 6868 has carbon fixation pathway? So
Which carbon fixation?
with carbon fixation, okay. So we have a genome, like a whole genome, but we did not look into this, like in the all. So I think with that information, we can answer your question. But the previous publication, the one that published it, it has a Rubisco. Yes. Your question?
Yes, from what I understand, like the earlier oceans on Earth, they were like chemically different because of the different redox state, like iron was like a different form. So in these like oxygen minimum zones, do we see like a different chemical makeup than we do in like oxygen rich zones? And like, how does that differ? And do you know?
So the question is, this chemical composition of the oxygen minimum zone is different from oxygen rich
Yeah,
compared to the early ocean?
yeah, I think you're getting at it.
Okay, yes. So, from what we know so far, the composition of earliest ocean, let's just focus on the Mesoproterozoic ocean, where it's closely resembled to oxygen minimum zone. So the model has been shifting a lot in the past 20 years. I think the original model proposed in 1998, where the upper layer is rich in hydrogen sulfide, but as more data collected, these model kind of shifted more heterogeneous ocean, but we still have a pocket at the mid depth where it's enriched in hydrogen sulfide. And the deep ocean, it's just a ferruginous ocean, which means it's enriched in iron. But otherwise, modern ocean, the oxygen go very deep into deep ocean. But if you're interested about deep ocean oxygenation, you should come to my general exam on January 22, which I will explain when the oxygen get oxygenated at deep ocean. Yes. But the deep ocean, the like deep, that's the difference.
The diagram doesn't show the oxygen minimum zone.
No, this is like a general depiction of
Yeah.
like overall ocean. Yes. We still have like, we have pocket of it, but I think the proportion of it is still like less. Yeah. Yes.
Like volume of these oxygen minimum zones, like how big are they?
Right now.
Yeah.
So the question is like how big is the volume of oxygen minimum zone right now? I don't know exactly. I could just estimate from this pocket, I guess. But yeah, so these are the oxygen minimum zone is like the upper layer is still oxygenated, but the mid-depth layer are enriched in hydrogen sulfide, and the lower one is still rich in oxygen. So it's like a pocket of mid-water. So yeah. Yes.
So when you were culturing your microbes, do you need to maintain an agnostic environment for them? If so, how do you do that?
No, I have a question of, oh, yes. So he's asking, do I have to maintain the anoxic environment, basically no oxygen in my culture? No, it seemed like they can grow in the presence of oxygen also. And that could be, might be like constrained because we can do it, but it's gonna be harder to culture it in low oxygen present. But we try to maintain by like a cap the bottle. But again, it's not a purely no oxygen environment. Yeah. I see hands somewhere else. Yes. No, behind.
So for these microbes, what were they,
UVA. UVA
What
is
did they- For them to metabolize, reduced or intermediate redox states of sulfur, someone has to be reducing sulfate to get those intermediate or reduced species into the water in the oxygen minimum zone. So out of all the organisms you cultured, did you come across any sulfate reducers that would supply these more reduced sulfur species for your target organism?
Oh, it's a hard question. So, Roger asking like, for UVA to metabolize sulfide in the water column, it need another bacteria to reduce sulfate to sulfide. And whether I enculture any those microsulfate producer in my culture, I don't. The answer is I don't know, because when I have the sequencing, I could have tried to look into that, or what do they do, but I did not. So, but I believe there are probably some to like maintain the balance. Yeah.
All right, well, I think we've hit the top of the hour, so why don't we thank Mac again for a fantastic talk?